I'm a geologist, but what we think about a lot in the lab that I work in is early life. And I'm not actually going to talk about the research that we're doing right now. What I wanted to do instead was sort of lay out some of the ways in which we used to do research that lead to a fundamental paradox that has inspired a lot of the research that we're doing now, okay? So here's one of the most iconic images in modern biology, probably the so-called tree of life, a illustration of relatedness of different organisms. There are the three major domains of life up there, the eukaryotes, archaea, and bacteria. And what we're interested in doing in my lab is trying to figure out how this tree of life grew. That is, when did the earliest branches, the most deeply rooted branches, first appear? So I want to lay out the recipe that we use in order to try and do this. Okay, so the first ingredient in this recipe is you have to identify an evolutionarily ancient, that is deeply rooted, form of life. We use sulfate-reducing microbes. These are bugs that instead of breathing oxygen, they breathe the sulfate molecule. They oxidize organic carbon with the sulfate molecule, producing carbon dioxide and also that rotten egg smell, hydrogen sulfide, okay? So the second ingredient that you need is you need to calibrate the biological signatures, the biosignatures that these microbes produce in the lab. And what we do is we grow the bugs in the lab and collect the hydrogen sulfide that they produce. This hydrogen sulfide in nature typically is very reactive towards iron and is preserved in rocks as fool's gold or pyrite. And what happens is that these organisms are so exquisitely tuned that they can tell the difference between a sulfate molecule that has sulfur-32 isotopes versus a sulfate molecule that has slightly heavier isotopes, sulfur-34, 2 extra neutrons, and the bonds that those heavier isotopes form with their surrounding oxygens are a little bit more energetic and a little bit more difficult to break. And so these sulfate-reducing microbes, they say, hey, let's go for the easy food. And the H2S that they produce is enriched in this sulfur-32. So what we do is we look at pyrite in rocks and we measure the isotopic composition of that pyrite looking for this biosignature. So the third thing that you need is really, really old rocks. Fortunately, there's an abundance of these in Canada, some of the oldest in the world. The oldest in the world were discovered by some of my colleagues over in EPS, 4.3 billion years old, okay? So the age of the solar system is 4.56. These are a couple 100 million years younger than that. And so what we do is we go into these rocks, crush them up, look for the isotopic composition of sulfur in these rocks, and try and say whether or not these sulfate-reducing microbes were around metabolizing at this time. And then we have a way to date that early branch on the tree of life. So there's the recipe laid out, okay? Really, really old rocks, a calibration made with modern organisms, and identification of an ancient form of life. There's a fundamental paradox in using this approach. I'm not going to tell you what it is. You all need to think about it. That's my secret question. And it's led to the new research program.
Can't be a secret, you just told us.
I know. Sorry. I had to give them a little bit of time. So this led to the new research program that we're doing in the lab now. So just a little bit of advertising before I close. Some colleagues of mine and I have designed 3 new courses, field courses for looking at Arctic field studies. These are going to be offered May, June of this year. 6 weeks of field work in a high Arctic setting, 9 credits. Awesome stuff. Hans Larsen over here and I are going to be co-teaching the Arctic Geology class. There's some ATOC colleagues, Bruno Tremblay, who will be doing Arctic climate, and then Wayne Pollard will be doing Arctic geomorphology. A second opportunity for undergrads that I wanted to mention is the Canadian Astrobiology Training Program. This is something that's run inter-faculty at McGill and inter-university, and there are undergraduate summer research fellowships available. You can apply at that website. Thanks.